Elegans by Bradley Hamza, Austin Veith, Haley Keaty, and Jasmine Taylor. Welcome to the Nematode Wildlife Tour. In this tour you will see numerous C. elegans in various stages of life along with the famous C. elegans from the development arrest experiment. C. elegans are unsegmented pseudocollomates that lack both a respiratory system and circulatory system. They exchange their gases directly through their bloodstream through their skin. C. elegans have four larval stages separated by molts, which are the shedding of the skin to allow new growth. C. elegans live very comfortably in temperate soil with high nutrient availability. Many factors can affect the growth of C. elegans, like high population density, high temperature, and reduced nutrient availability. Alright class, this picture shows what a typical C. elegans looks like, and also the different components and parts that make up the entire C. elegans. Next class, this is a typical illustration of the life cycle of a C. elegans. As you can see here, it displays the L1, L2, L3, and finally the L4 stages of the C. elegans lifespan. And this is all very interesting and ties into what you're about to see next. And now class, for what you've all been waiting for, the C. elegans from the famous development arrest experiment. Here you will see a population of C. elegans that are approximately 12 days old. Their average lifespan is about 4 days, so 12 days is pretty old. These C. elegans have been through a lot, so take caution around them. In this experiment, they tested a whole population of C. elegans and various food availability. Let's see what they have to say. Now class, these C. elegans are pretty old, so we'll be visiting them in the old worms home today. Well, back when I was in a young L1, things were very different. They took my thousands of brothers and I away from our home and put us in a laboratory. We lived very comfortably until we reached our L2 birthday. They separated about half of us and took our food away. The other half still got their normal food and continued their lives, but we were left to waste away with no food. But surprisingly, we turned out to be fine and actually lived longer than our brothers that had food. So this is very interesting and all, Professor, but is this story actually true? Why yes, certainly, very true. Adam Schneider, Ryan Baugh, and David Sherwood conducted an experiment on these C. elegans and determined their growth pattern at various larval stages with varying food availability. But Professor, how can they still survive without food? Well, class, they took away their food at various times from the L2 to the L4 stage. They found that the growth arrested for most of the individuals when food was taken away at L2. They also found that growth seemed to be regulated by checkpoints and that after an individual got past a certain checkpoint, they continued growth until the next checkpoint where nutrient availability was reevaluated. But Professor, we're still confused. How does that happen? Well, class, the insulin signaling pathway is a key regulator of growth in response to nutrient availability. The insulin signaling targets DAF16, which is a transcription factor that plays a role in L3 and L4 arrest. A transcription factor is simply any protein that binds to a regulatory site on DNA and affects the transcription rate of that gene. The transcription is essentially how often the gene is read by other proteins and transcribed into mRNA that eventually turns into other proteins. When insulin is present, DAF16 is inhibited, halting arrest and continuing growth. Therefore, when insulin or food is present, the C. elegans die more readily because they grow and continue through their life stages faster. When food isn't present at these checkpoints, they determine that growth is halted in a vast majority of these C. elegans and that they can actually live longer at an arrested development state. Wow, that is so interesting. They must be like superheroes. No class, just some normal C. elegans.